Okay, so moving on to the last part of the male reproductive system, that is the hormonal control of testicular function and spermatogenesis via the HPG axis cascade. HPG is the hypothalamus pituitary gonadal axis. As we know, at puberty, hypothalamic cells increase the secretion of gonadotropin releasing hormone, that is GnRH. This GnRH in turn stimulates gonadotropins in the anterior pituitary to increase the secretion of luteinizing hormone and follicle stimulating hormone. The LH in turn stimulates Leydig cells to secrete the hormone testosterone, which is the principal androgen in males. In target cells, that is external genitals and prostate, testosterone is converted to dihydrotestosterone by the enzyme 5-alpha reductase. Testosterone and dihydrotestosterone both bind to the same androgen receptors found in nuclei of target cells, and the hormone receptor complex regulates gene expression, in turn producing various physiological effects. FSH, at the same time, FSH acts indirectly to stimulate spermatogenesis. FSH along with testosterone act on Sertoli cells to stimulate the secretion of androgen binding protein. This androgen binding protein is responsible for the actions of testosterone, that is ABP binds to testosterone, whereby testosterone can stimulate the final steps of spermatogenesis in seminiferous tubules. Once enough spermatogenesis has occurred, that is once enough sperms have been generated, the Sertoli cells release inhibin, which inhibit FSH secretion by anterior pituitary. If spermatogenesis is occurring slowly, then less inhibin is released and more FSH will be secreted, increasing the rate of spermatogenesis. A negative feedback mechanism also regulates testosterone production. When the testosterone concentration in blood increases, it inhibits the release of gonadotropin releasing hormone by hypothalamus. Gonadotropes in anterior pituitary secrete less luteinizing hormone, whereby luteinizing hormone concentration in blood decreases. The action of LH is reduced and Leydig cells in testes secrete. We can study the functions of testosterone and dihydrotestosterone. So both the hormones, they are analogs, and they principally act on androgen receptors, which are mainly nuclear receptors. So the hormones, they mainly help in prenatal development. In prenatal development, that is just before birth, testosterone stimulates the male pattern of development of reproductive system ducts and the descent of testes. That is around 7th month, the testes will descend towards via the inguinal canal, dihydrotestosterone, and then stimulates the development of external genitals. Then the development of male sexual characteristics, that is at puberty, the hormones will bring about development and enlargement of the male sex organs and the development of masculine secondary sexual characteristics like muscular and skeletal growth that results in wide shoulders and narrow hips. Then facial and chest hair will be developing, thickening, the skin will be thickened, there will be increased sebum secretion, oil secretion, which may lead to pubertal acne, and there is enlargement of larynx as well as consequent deepening of the voice. Then the other functions are development of sexual function. Androgens mainly contribute to the male sexual behavior and spermatogenesis, and also increases libido in both males and females. That is, it stimulates the sexual activity. Then the hormones also help in stimulating anabolism. The androgens are anabolic hormones, that is, they stimulate protein synthesis, which can lead to the development of heavier muscle and bone mass of most men as compared to women. So in short, a sperm cell, we can just summarize the complete journey of a sperm cell, starting from the trigger, that is GnRH, LH and FSH, initiate the cascade. Leydig cells produce testosterone and Sertoli cells produce ABP. Then the spermatogenesis occurs over 65 to 75 days inside the seminiferous tubules, and spermatogonia transform into haploid mature spermatozoa. Transfer into haploid spermatozoa. Then the maturity occurs in while passing through the ducts. They acquire motility in the epididymis over 10 to 14 days, and then they are transported through the vas deferens. Then via their during the course of their transport, the semen is also secreted from the vaginal. The semen is also secreted, which is contributed by the seminal vesicles, prostate gland, and the bulbourethral glands. And finally, on ejaculation, ejaculation, there is an expulsion of 2.5 to 5 ml slightly alkaline semen to the penile urethra, which is engineered for survival and fertilization, survival of sperms and fertilization of ovum in the female tract. So that's all about the male reproductive system. The main points that we are highlighted over here is the different organs, glands and the gonads that are involved in the male reproductive system. Then what are the functions of each of the individual organs and glands. Then we have studied about the structure of sperm. Then we have studied how spermatogenesis occurs in the testis, and finally we have learned about the different hormones that are involved in the male reproductive system and how they regulate spermatogenesis as well as other male characteristics.